Okay, this is lecture 2. We'll be talking about the introduction, scope, and history of parenteral drug products. Again, parenteral drug means what? The injectable drugs. Okay, so we'll continue discussion on that as well. So the history, first of all, history of parenteral drugs. So since when people started injecting the drugs into human body? That's the question, right? It started as early as 1656. Some scientists experimented injecting drug into human body. Of course, before testing that into human, they tested on the animal. So 1656, some scientists tested, Christopher Wren tested injecting some opioid into dogs and see if he could have some kind of opioid effect from dog. Okay, so he developed some kind of crude syringe, like a structure, like animal blood and goose kill, and through that crude injection device, he tried to inject some opium to the drug intravenously. Kind of crude experiment. Okay, and after that they tested that in 1660 on a human patient to minimize the pain. Of course, tried to inject some kind of same opium to a patient, but unfortunately the condition was not good, so that patient ended up dying because of the infections, because microbial infection and pyrogenic infection. We'll talk about what pyrogens are. The patient died instead of getting some help through the painkiller, the opioid. Okay, but however, the experiment continued. So in the 1700s, about almost like 1800, 1796, the vaccination study. Edward Jenner studied the intradermal injection of cowpox virus. Okay, cowpox is different than chickenpox that happens in human, but there's a little bit of difference in the structure and then infection mechanism. So that is the beginning of the vaccination, and by getting this cowpox vaccination, people developed immunity against the chickenpox. Okay, so that's 1796, and then 1831, the first intravenous therapy. So giving the saline injection through the blood vessel. Okay, that's studied. So they developed a saline solution, sodium bicarbonate mixture. Okay, and that mixture of water is given to a cholera patient, because cholera patient normally has severe diarrhea, so they lose a lot of water from their body. So that's how they die. So the intravenous IV treatment studied in 1831. Okay, so that's 1800. In the mid 1850s, the first hypodermic needle syringe was developed. Okay, so hypodermic under the skin needle syringe was developed and used for subcutaneous injection. Remember that diagram I showed you, different way of injecting injectable drugs. So there was an IV injection and then subcutaneous injection. Okay, that's the start. But even that time, people didn't understand the contamination of microbials that can happen through injections. So a lot of people got sick and died, and they wondered what's happening. So some scientists developed the germ theory. Okay, a lot of disease may be caused by microorganisms that are invisible, but they may go into human body, causing infections, right? So that was the prevalent thought about human disease at that time, 1860. Although it's not all the diseases are caused by that, but a lot of infections and disease at that time, people die from those, like pneumonia, for example. A lot of people died at the time. Okay, so they understood, the importance of clean condition and the sterilization of your drug product and all those things. So after understanding that, they thought about ways to clean up, ways to sterilize your drug product, of course. So 1884, the first use of autoclave, it's a steam cooker, right, was developed. So before you give the injection of a drug, that drug should have been sterilized. That's what they developed and start doing it. Okay, Chamberlain. And then not only the heat sterilization method, there are different ways of sterilizing your drug, of course, right? You can use gas as well if you are dealing with your medical device. Okay, gas may be a better way of sterilizing. So gas sterilization method was developed 1859. Okay, and then they started using formaldehyde gas, which is very toxic, cancerous gas. And then they also started using ethylene oxide for gas sterilization method. And then later on, of course, they came up with another method that is radiation with UV light. Of course, strong UV light can kill microbials as well. So they started using it as well. And besides this, there's another way. What is that? That filter sterilization method. Okay, that was started being used in 1850 as well. So different ways of sterilizing drugs. So because they understood purity of your drug is very important. Okay, and then 1923, they understood besides the living microbials, there are something else that can cause the problem. Okay, they understood there's something called pyrogen or endotoxins that can cause pyrogenic reaction, which can kill patient. Okay, so pyrogen endotoxin, probably you heard this term before, and if you haven't heard, please understand what that is. Okay, so pyrogen, another word for them is endotoxin, are actually not the toxins that are produced by bacterial cells. Exotoxins are actually special toxins that are made by the bacterial cells and they're released, but endotoxins are not toxins. Actually, those are broken debris of the cell wall of gram-negative bacteria like E. coli. Okay? And those are working as toxins when they come into human body. Okay, then what is this? There are rabbits. Okay, so this person is injecting something into the vein of the rabbit ear, right? So this is actually the endotoxin testing that you can do when you make a drug. How do you know if you don't have a lot of endotoxin in your drug, the final product? You can test it by giving an injection to the vein of a rabbit and check the temperature, body temperature of the rabbit, and see if it causes any pyrogenic reaction or not. That was the animal testing that had to be done as part of the requirements. But a lot of people thought that is very inhumane, so we don't do that anymore. Instead, we use something else. So again, this is the endotoxin reaction. What's happening? So in your body, in your immune system, there are these cells that can kill infectious bacterial cells or viruses. Those are macrophages, right? They can engulf bacterial cells that are coming into your body and then digest them and kill them. That's part of your defensive system. But when they kill them, they digest them, the bacterial cells are broken down into small bits of molecules, right? Among those, if the cell wall materials are broken up, they can be released from these bacteriophages. And if they are released, then they'll be floating around through your blood vessel in blood, and some of them can go into your pituitary gland and then start the hypothalamus reaction, which is releasing a lot of hormones and the signals. So your immune system will be hyperactivated, and the part of the hyperactivation is causing high fever, and that high fever will be uncontrollable. So people would die from the high fever condition. That's a septic shock or sepsis caused by the human body, and then a patient may be dying because of that. So that's the dangers of the endotoxin. Okay, so what I'm saying is, if those endotoxins are present in your drug product, actually you can kill your patient, because you have to inject your drug product if your drug product is parenteral drug, right? So you have to make sure that your parenteral drugs do not have endotoxins in them. Okay, that's very important. So then, instead of doing this inhumane rabbit test, then what else can you do? So these days, drug manufacturers are using something called LAL assay. LAL stands for limulus amebocyte lysate assay. Okay, then what is that limulus amebocyte lysate? So actually, limulus amebocyte are these. Okay, so what are these? The blue blood that are taken from, what are these? Horseshoe crab. crustaceans living at the bottom of the ocean. Okay, and you can collect some blood from them, and their blood cells have a special activity. They can bind with the endotoxins. Okay, and once they bind, they coagulate. Okay, so the coagulation is the key in this assay. If there are a lot of endotoxins present in your sample, they'll be coagulating with LAL, limulus amebocyte lysate, and then they may form a network, gel clot, or they may form some color. If you put some pigments with this LAL, you can see the development of color. So that's the basis principle of this LAL assay. So this is presenting you with a very detectable assay as well as very quantifiable assay method. So instead of using this inhumane method, and nowadays you are using the LAL assay to detect the amount of endotoxin present in your drug product. Okay? Understand the importance of endotoxin and how you can detect that in your drug product using an LAL or the rabbit assay. Okay, let's continue. So 1938. The Food, Drug, and Cosmetic Act was passed by the Congress, United States Congress, and that was passed because they felt the need of that law. Okay, so what is that? What was the need? There was a huge incident where about 107 people died all over the United States at that time. The incident was caused by this medication, which is antibiotic. Okay, it's sulfonamide. Sulfonamide is an antibiotic that is prescribed by doctors when people get sick with pneumonia, so it can treat the patients with pneumonia. But the thing is, some pharmacists decide to formulate this sulfonamide with something flavorful. Okay, what they used for the formulation was diethylene glycol. Some of you know what diethylene glycol is, right? That's the coolant that goes into your car engine. It's a coolant, and strangely, that is very flavorful. It's sweet and flavorful. So some pharmacists thought, oh, if they can mix up this sulfonamide, the antibiotic that's very bitter, with this flavorful excipient, and then they can give to the especially kids, small age children. Okay, so because of this toxic substance mixed with the drug and given to the kids, a lot of children died at the time, 1938. So all over the United States. So there was the federal investigation that's happened, and they understood what happened. And then after that, they decide to set up a law, federal law. That is the beginning of the Food, Drug, and Cosmetic Act. And based on that Food, Drug, and Cosmetic Act, so the agency, government agency called FDA was developed and established. Okay, so the federal law was started in 1938, and then the federal agency of FDA started after this huge incident. Okay, so this FDA from that time on is reinforcing that drug manufacturers should prove that what they're manufacturing should be the safe drug that's not going to kill patients like this sulfonamide. Okay, so that's the beginning of the FDA. In 1940 again, penicillin started being used. Okay, so super drug, antibiotic, and used a lot during the Second World War. Okay, and then freeze drying was started being used, developed and used, because when the Second World War was started, the war was happening in the Pacifics and the European countries, but American soldiers were dying there, so they wanted to come up with a way to send plasma to the long distance, and for that this freeze drying method, lyophilization method, was invented and developed. Okay.